In this section, we'll be looking at protein structure. We'll be looking at what determines the three-dimensional structure of proteins and seeking to understand the intramolecular interactions that promote these particular structures. In this section, we will discuss protein structure and we'll be focusing on primary and secondary structure. So primary structure is the identity of the atoms within a protein, so the chemical identity of the protein and the order of the amino acid sequence. And secondary structure involves the formation of hydrogen bonds between different amino acids. And this determines whether the protein can form alpha helices or beta sheets, which we'll cover in this section. Now this is a structure of a protein from a virus that infects bacteria. This organism is called a bacteriophage. Now this isn't important, but we're going to be focusing on this protein as an example, and we're going to be looking to understand what determines the three-dimensional structure of this particular protein. Now proteins have quite a rigid structure. They are flexible, but not infinitely so. So we'll be looking at the interactions that stabilize and hold this particular structure together, giving it the shape that it does, and why this shape is important in the context of its function. Proteins have 4 different levels of structure, and each of these involves a different set of intramolecular binding interactions or even intermolecular binding interactions that help determine the overall shape of the protein. So the 4 levels of structure referred to as primary, secondary, tertiary and quaternary, and we're going to go through each of these in this section. Now the first level of structure is primary structure. This is simply the order of the amino acids in the protein molecule. So this is how the amino acids are joined together and linked through peptide bonds. Now all amino acids are linked together through a peptide bond. Now a peptide bond has a carbonyl group, so a carbon double bonded to an oxygen, followed by an NH group, which you can see here. Now the fact that the peptide bond has these 2 groups next to each other helps to determine the shape of the protein. And the reason for this is that the peptide bond, because of a process called resonance, is planar. It cannot freely rotate. So why does it do this, or what causes this? So this is a peptide bond here with our NH group and our C double bond O. And what happens is that the nitrogen can donate its lone pair of electrons into the nitrogen-carbon bond. This then pushes the electrons from the carbon-oxygen double bond up into the oxygen to give the oxygen a negative charge and to form an oxygen-carbon single bond. We then have a carbon-nitrogen double bond and the nitrogen has a positive charge. Now this process is called resonance. And this process is constantly happening at every single amino acid within the protein. So we are getting continuous interconversion between the 2 forms. And this means that some of the time the peptide bonds have a double bond, and some of the time they don't. But most of the time they do. And this means that the peptide bond cannot freely rotate. So this means that the peptide is planar. Now, amino acids typically adopt the trans conformation, so they will adopt this form, and they do not typically adopt the cis conformation, with the exception of one amino acid. So it means that this resonance not only locks the peptide bond so that the peptide bond is planar, but also locks it into a trans conformation, like this. And so this order of the amino acids is actually very important to protein structure, as we'll see in a minute. Interactions between the amino acids also help to determine the three-dimensional structure of the protein. So let's go back to the structure of the protein from our bacteriophage. If we now colour the atoms according to their type, so we have carbon in green, oxygen in red, nitrogen in blue, and sulfur in yellow, what we can see is the chemical identity of all the atoms within this protein. Now hopefully you can see some regular patterns in there, and the reason for this is that all amino acids have essentially the same structure, apart from the structure of their side chain. And it's the structure of their side chain that determines the interactions that an amino acid can have with other amino acids. And we'll look at that in a bit more detail in a few moments. So let's have a look at the chemical structure of amino acids. Now there are 20 naturally occurring amino acids, and this is the structure, or these are the structures of amino acids which contain hydrophobic groups. Now first of all, if we look at the sections in blue, these are the same for every amino acid. So the structure in blue is conserved with every single amino acid in this series. What changes the properties of each of these amino acids is the side chain, which is shown in black. Now the side chains of all of the amino acids shown on this slide are amino acids which contain hydrophobic groups. So these are groups that do not like to be around water. Now I just want to stress that you do not need to memorize all of the amino acids. You don't need to learn how to write these down or how to draw them out or anything like that. But you should be able to identify some of the properties of the amino acid from its structure. So for example, if you were asked to have a guess as to what properties you think phenylalanine would have, you could look at its side chain and see that there's no heteroatoms, there's no OH groups or anything polar, and there is an aromatic ring. And this would tell you that the lack of polarity in this system would mean that this amino acid side chain is hydrophobic. Now in a moment we'll have a look at other amino acids containing hydrophilic or polar or charged side chains. But these are the 8 which contain hydrophobic groups. Now these are the structures of the amino acids containing polar groups. So what you'll see here is chemical features like alcohol or OH groups. You'll also see carbonyl groups, amine groups as well, and thiol groups. So these are SH groups rather than OH, which you get for hydroxyl groups. Now all of these amino acids have polar side chains. And again, you should get used to looking at the structures of the amino acid side chains and trying to identify what properties they may have. Now, of course, this is also important when we're considering what interactions those side chains could have with drug molecules. So, for example, the OH group on serine. An OH group can be a hydrogen bond donor or an acceptor. The same for the OH group on tyrosine or for threonine. And so the OH groups here could act both as hydrogen bond donor or acceptor. And this means that if we know we have these amino acids in close proximity to our target binding site, that we could make a molecule capable of hydrogen bonding to those chemical groups. Now these are the amino acids that contain ionizable side chains. So we have lysine, which can hold a positive charge, arginine with a positive charge, and histidine with a positive charge. We also have aspartate and glutamate, which can hold negative charges due to them having a carboxylic acid within their side chains. Now again, if we knew that we had these amino acids in close proximity to our binding sites, we could produce a compound that has a functional group capable of forming an electrostatic interaction with one of these side chains. But we would need to know which side chain we have in order to work out whether we put a group on our drug molecule that can have a positive or a negative charge. But knowing what groups we have means that we can choose the properties of our drug molecule and incorporate groups to be able to bind to these types of side chains. This is a short peptide sequence called metenkephalin, and it's a natural painkiller that's constructed from 5 amino acids. Now I'm going to use this to illustrate how you should read off an amino acid sequence. Now this method is the same as whether you're reading off a short peptide like this or a very, very long protein. Now you always start at the N-terminus or nitrogen-containing terminus, and you read the sequence of amino acids off until you get to the C-terminus here. So the sequence of amino acids in this chain would be tyrosine, glycine, glycine, phenylalanine, methionine. It's important you don't try and read this in the other direction because that would give you a different amino acid sequence. And so you must always remember to read from the N-terminus to the C. So we've had a look at the primary structure of proteins, so let's move on now to look at the secondary structure. Well, in the secondary structure of proteins, we can form 2 different types of structure, either an alpha helix or a beta pleated sheet. And these form due to hydrogen bonds between the peptide bonds that can occur when the amino acid chain folds up. Now, secondary structure does not involve any interactions between the protein side chains, between the amino acid side chains. It only involves hydrogen bonds between the peptide bonds. Now the first secondary structural feature is the alpha helix. Now we can see here this forms a helix within the protein. Now if we look at the peptide bonds, you'll see that the carbonyl group, so the carbon double bond oxygen, acts as a hydrogen bond acceptor for the hydrogen that is attached to the carbon in another amino acid. Now this acts as a hydrogen bond donor. Now this is an example of an intramolecular binding interaction because the interactioning is happening between functional groups in the same protein molecule. Now the side chains of the amino acid actually protrude or project at right angles to the axis of this helix. So if the helix is going up, the side chains will project out around the helix, and this minimizes clashes between the atoms in those side chains. Now this alpha helical structure is one of the main ways in which a protein folds up to get its secondary structure. Now the other way it can fold up is to form a beta sheet, which we'll have a look on the next slide. Now this is a beta sheet. Now this is also held together by hydrogen bonds between the peptide bonds. So we can see here again, the carbonyl group in one peptide bond is acting as a hydrogen bond acceptor for the hydrogen attached to a nitrogen in another peptide bond. And this group here is acting as the hydrogen bond donor. Now, folding into this motif, into the beta sheet, allows the protein chains to form layers, and the residues all stick out above the beta sheet, pleated sheet, or below the sheet. So you can see one there and another one here. Now these can be parallel or anti-parallel in orientation. Now what I mean by this is the amino acid strands can either go from left to right in both cases, so they're going in a parallel direction, or you could have one going left to right and the other one going right to left in an anti-parallel orientation. And this is because the structural flexibility of the protein backbone allows the chain to fold or to turn and go in the opposite direction. So let's have a look at this in a little more detail. So we can form both a parallel and anti-parallel beta sheet. So a parallel beta sheet is where the directionality of the 2 peptide chains goes in the same direction. So remember, when we looked at reading peptide chains, we always go from the N-terminus to the C-terminus. And that is what we mean by directionality here. So the top strand in this image is going from left to right, so the N-terminus would be on this side, and the C-terminus on the right-hand side. And that would be the same for the strand below as well. Now this can form, again, hydrogen bonds between the carbonyl groups and the hydrogens of nitrogens in peptide bonds in the amino acids. Now we can also form an anti-parallel beta sheet where one strand goes in one direction and the other strand goes in the opposite direction. Now this is because we can form something called a beta turn, which is what the beta turn looks like. Due to the flexibility of the strand can fold over on itself and enable the direction to reverse. Now again, I just want to highlight that the hydrogen bonds are between the backbone groups. No side chains are involved in protein secondary structure, and therefore the secondary structure of the protein is not determined by the identity of the amino acids. All of the amino acids equally have the possibility of forming these hydrogen bonds. So let's return to our diagram of this protein. And here, of course, we're looking at the primary structure of the protein. So we're looking at the atoms labelled according to type. But we can start to trace the backbone and add in some of the secondary structural features. So here you can see the backbone of the protein. And if we colour code this, what we can see is the alpha helix in red and the beta sheets in yellow. Sections in green are not ordered, so they're not held together with a secondary structure. But we can see this protein has two alpha helices and a single beta sheet that has been folded over. In the next section, we will discuss both tertiary and quaternary structure as applied to protein molecules.